Hey guys, we're going to talk about muscle tissues today and kind of how a muscle contracts and what's inside of a muscle. Okay, so before we learn all the muscles of the body, it's really important to know kind of how a muscle works. And before we talk about that, we need to talk about what's really inside of a muscle. So a muscle, if you think about it, is just a bunch of tubes within tubes within tubes. And these tinier tubes shorten, and that causes the whole muscle to shorten, which pulls on bones and generates force and motion. So if we were to draw this out, let's imagine you've got some person like flexing his bicep, right? So we use the bicep as an example. The bicep is obviously a whole muscle. Okay, so here this person kind of flexing his bicep, right? And this bicep right here, that is the whole muscle. Okay, so right here, circle this, that is the whole muscle. That's kind of the first level of our organization. But let's imagine that we kind of cut this muscle in half. Wouldn't be pretty painful, but let's cut this muscle in half so we can see what's inside. And if we do that, we'll notice that this muscle is going to be composed of a series of smaller tubes that make up the whole muscle. These smaller tubes kind of look like this, right? There's going to be a series of smaller tubes. If we take one of these tubes and expand it out like this, okay, so here's one of those tubes, we expand it out like that, we'll notice that each one of these tubes that make up the whole muscle, those are called fascicles, okay? So this is one of those tubes that is located inside the whole muscle. This is called a fascicle. F-A-S-C-I-C-L-E. Now the whole muscle and the fascicle are both wrapped in a layer of connective tissue. The connective tissue that wraps around the whole muscle, this is going to be a layer of that. This is green layer I kind of drew right here. That is called epimysium. Okay, and then inside of the whole muscle, each fascicle is going to be wrapped with another kind of layer of connective tissue, which I'll draw right here. That's called perimysium. Okay, and the perimysium wraps together each fascicle. It's also kind of located between each fascicle. And that kind of just holds these structures together. Now each fascicle is going to be composed of a series of smaller and tiny little tubes. There's a bunch of little tubes in each fascicle. Each one of these little tubes is actually a single muscle cell. So now we're at the level of a single muscle cell. Another name for a single muscle cell is called a muscle fiber. Okay, so if I were to expand out one of these guys, which I'll do right here, a single muscle fiber is going to look like this. Okay, so imagine we just took one of these little fibers and expanded them out. He would look like that. Now, again, a single muscle fiber is a muscle cell. Muscle cells are kind of unique in their shape. They are long, right? They're much more longer than your typical cell. They have multiple nuclei, so we'll draw like multiple kind of nuclei that are distributed along this very long cell. Okay, and they're just shaped like little tubes. Okay, they are kind of covered in another layer of connective tissue called endomysium. So we'll have like some endomysium that kind of wraps around this. But most importantly, what wraps around that muscle fiber that's very close to the cell membrane is a special type of nervous tissue called sarcolemma. So that's what I'm going to draw in this pink kind of, these pink lines represent the sarcolemma, which is a special type of nervous tissue it's nervous-like tissue that carries an electrical signal throughout the muscle and stimulates each one of these fibers to contract. All right, so we'll talk about kind of how that works specifically later on, but this pink covering is called the sarcolemma, and that's located right here. And then this tube right here, that is an individual muscle fiber, which is the same thing as a muscle cell, okay? All right, perfect. So inside the muscle fiber, that's also composed of a bunch of little tubes. Now, just to put this into perspective, we're at the subcellular level now. So we're inside the cell membrane and we're looking inside of a single cell. This muscle cell is going to contain all the regular organelles like nuclei, like here, mitochondria, lysosomes, all that. But it's also going to contain these tiny tubes. Each one of these tubes is called a myofibril. If we expand one of these myofibrils out, it looks like this, okay? That's one of these myofibrils. Now these myofibrils, these are subcellular components of the muscle, okay? And this guy, so this is a myofibril, he consists of not little tubes, but of a grid of proteins. So this guy is going to consist of a grid of proteins. All right, I'm going to try to draw this out the best I can. The grid of proteins that kind of make up this myofibril. Right, it's this grid of proteins, kind of looks like this, right? Kind of make it look like a grid. And you know, if we cut it in half, we'll see that that grid exists throughout the entire diameter of this myofibril. Okay. Now, essentially, what happens is that when this muscle contracts, this grid of proteins gets shorter. When that happens, all the myofibrils get shorter, which causes all the muscle fibers to get shorter, which causes all the fascicles to get shorter, and eventually the whole muscle gets shorter. As the whole muscle gets shorter, it's attached to parts of the skeleton at least, and that's going to pull on those parts of the skeleton and induce motion about a joint. And that's really kind of how a muscle works. Now, if we were to zoom in on a little piece of this grid, let's say we zoomed in on like a little piece of the grid right here, and expanded it out so we can see what's inside, we'll see that this grid is actually composed of two major types of proteins. We have these proteins in blue, which create this kind of rectangular, these repeating sets of rectangular shaped proteins. Right, so these guys in blue, these are called the thin filaments, or actin, A-C-T-I-N. And then these red proteins, or the pink proteins, those are the thick filaments, they're thicker, and they're called myosin. M-Y-O-S-I-N. Okay. Now, what essentially happens is that these myosin proteins, they have these special, like, little appendages. They're called myosin heads that extend out. It looks like a little, I don't know, weird looking caterpillar or something like that. But these appendages are extending out in all directions, kind of toward, out of the board, up, down, not just to the side. And what happens is that the myosin will use these heads when the muscle contracts to reach out. These heads will grab on to actin. Okay, so these heads reach out, grab on to actin, like that. The heads on this side of the myosin molecule, they reach out and grab onto actin and they pull that way. These guys pull this way. These heads reach out, grab onto actin, they pull this way. These pull this way. Not just this myosin molecule is doing this, but all of them are in the entire grid. And that causes this grid to shorten, right? And if you think about it, if all of these little units are doing that, the whole myofibril shortens, the whole myofiber, the fascicle, and the whole muscle shortens, and the muscle contracts. Now, a little bit of important terminology. The name of one of these repeating units is called a sarcomere. So, one myosin molecule and all of the actin molecules that surround it, those are called a sarcomere. So I'll kind of circle one of these in green because that is a sarcomere. Okay. Good. Now, it's important to kind of talk about what really happens at the level of one of these myosin heads and how this guy is able to grab onto actin and pull. And so that's what we're going to do here. So we're going to really like zoom in on one of these little myosin heads that grabs onto an actin molecule, and that's what I've drawn here. So here is one myosin head relative to an actin molecule. So first thing that we'll mention is that right next, attached to, in the first step, attached to this myosin head, we're going to have a molecule of ATP, which is energy, right? Stored energy. Okay. Now, the first thing that will happen is that this ATP will hydrolyze, which means it releases its energy. And when it does that, it causes the myosin head to change shape, right? When proteins absorb energy, they typically change shape. Well, this myosin head is going to go from this kind of downwards position to a position like this, where he's reaching out. The ATP is hydrolyzed, but the components of the hydrolyzed ATP ADP, adenine diphosphate, and the phosphate that came off, adenine triphosphate split into adenine diphosphate, 2 phosphates, and an extra phosphate. So ATP turns into ADP and then extra phosphorus. They're still attached to myosin. Okay. What myosin wants to do is it wants to grab onto actin, but it turns out it can only grab on in very specific places. The places that it can grab onto are called binding sites. That's what these little circles represent. If these binding sites are available and myosin's right there, he will in fact grab on. In this case, that binding site's available, so myosin will grab on. Okay, so he reaches out and he grabs right onto that binding site. When that happens, it causes the ADP and the P to fall off of myosin. So now we have the ADP and the P. They're gone, they've fallen off. So P here, ADP, these guys have fallen off. And when that happens, there's nothing holding that myosin in this position, so he returns to his original position. This is called the power stroke. When he does this, because he's attached to actin, he's going to pull actin in this direction. So this guy is going to now pull, okay? And he has motion of both the myosin head and the actin molecule, okay? That's called the power stroke from this step to this step. So we could really label this as like A, B, C, and then D, okay? Now the only difference between D and A is the fact that ATP is here. So when an ATP molecule comes in after that myosin molecule has done his power stroke, it's going to cause that myosin head to detach. And this is going to continue to happen as long as ATP is available and these actin binding sites are exposed. Alright, now that we've talked about kind of how a single myosin head is able to attach and pull on actin, I want to talk a little bit more about how this process is controlled. Because this process in which this cycle where things go from A to B to C to D is called crossbridge cycling, and it's going to proceed as long as there's available ATP, which is always going to be the case, you know, the muscle's got plenty of ATP, and as long as these actin binding sites are exposed. Okay? This guy is going to keep attaching, pulling, releasing, attaching, pulling. If the muscle needs to relax, then one way to stop this cycle is by covering up these binding sites on actin. If that was the case, the cycle stops right here at letter B. The myosin head's ready to grab on, but he can't proceed to step C because those actin binding sites are blocked. This is how this is accomplished. In the relaxed state of the muscle, we've got actin, and we've got the myosin head, but there's two other proteins that exist in this situation. We've got this dark blue troponin, which is attached to a string-like protein in orange called tropomyosin. In the relaxed state, tropomyosin completely blocks the actin binding sites and prevents those myosin heads from grabbing onto actin. So when your muscles are relaxed, just completely relaxed, every myosin head and every sarcomere of every myofibril is ready to grab onto actin. They've just got nothing to grab onto, okay? Now, in order to expose those binding sites, calcium, the ion calcium, is sprinkled onto this sarcomere, and specifically this actin molecule. So here we're going to have a bunch of calcium that gets released onto the sarcomere when the muscle needs to contract. Calcium is going to bind to troponin, and when that happens, it causes troponin to change shape, and it's going to cause him to twist, kind of like a little switch. And now tropomyosin is going to get out of the way, which exposes these actin binding sites, or the binding sites on actin. With the binding sites exposed, that myosin head can reach out, grab onto actin, okay? That's going to cause the ADP and P to fall off, and we jump right into the crossbridge cycle. That's going to continue to happen as long as calcium is released. So, in a nutshell, when we tell our muscles to contract, a nervous signal comes from our brain, travels down a neuron, that causes the muscle to be stimulated, which ultimately causes calcium to be released onto the sarcomeres, exposing these binding sites, triggering crossbridge cycling. So I wanted to talk now a little bit about how a muscle is stimulated by a neuron or a nerve to fire. Okay, so we've talked about kind of how the cross-bridge cycling works and how a muscle contracts. If we zoom in and look at the level of the proteins like actin and myosin, but we haven't talked about what causes calcium to be released onto these sarcomeres, which triggers muscle contraction. And it's all about this neuromuscular junction. So this is the junction between a neuron, which is a nerve cell, and a muscle fiber. So here is the end of the neuron. It typically branches into a couple of different axon terminals. We have an axon terminal here, here, and here. This is the axon of this nervous cell or neuron. And here's a single muscle fiber. All these little black circles, those are all the myofibrils that make up that muscle fiber. So in order for a muscle to contract, it has to receive a signal from this neuron. It's regarding skeletal muscle, right? And if it doesn't, so a muscle should never really contract unless it receives the signal from a neuron. If it contracts without receiving that signal, it's either a spasm or a cramp. I mean, neither of those things are good, right? So in this case, what will happen is you'll have this electrical signal that is sent from the brain, and it travels down this axon, and it reaches the end of the axon in these axon terminals, okay? This nervous signal is then, it reaches a dead end. It's got nowhere to go. Well, when it reaches these axon terminals here, here, and here, that's gonna cause calcium to come into the axon from the surrounding extracellular fluid. So calcium is gonna come in. That's kind of the first step. And in orange, this is all calcium. So calcium is going to come into the axon terminals when this nervous signal reaches them. And that's going to cause something special to happen. Because inside these axon terminals, you have these little bubbles. They're little spheres that are made up of the same cell membrane that makes up the outside of the neuron. Well, these little bubbles or spheres, they're called vesicles. And these vesicles contain kind of tiny little chemicals called neurotransmitters. This neuron that stimulates the skeletal muscle, it contains the neurotransmitter acetylcholine. All right, so in green, this is all acetylcholine, which is abbreviated ACH. Okay, there's a couple thousand acetylcholine molecules in each vesicle. Well, when calcium comes in, it causes these vesicles to fuse with the cell membrane of the axon itself, the axon terminal. Okay. Well, when that happens, these little spheres, they're made up of the same material as the cell membrane that coats the outside of the neuron. Think of them as bubbles. It's like when two bubbles come into contact, they form into one bubble. So when calcium comes in, these vesicles fuse with the outer membrane. Okay, and when that happens, these vesicles then release the acetylcholine that they were containing into the tiny space that separates this axon terminal from the muscle fiber. That tiny little space right here, here, and here is called the synapse, and these molecules of acetylcholine diffuse across the synapse. Well, on the other side of the synapse, that's where we have our muscle fiber. The synapse is super small, it's like 30 nanometers in diameter, so they don't diffuse very far. Right? Well, on the other side of the synapse, that's where we have our muscle fiber. And if we remember, this muscle fiber is coated in this special nervous-like tissue called the sarcolemma. So everything in pink, that's the sarcolemma. And the sarcolemma has special receptors that will bind onto acetylcholine, which then triggers a new nervous signal to spread in all directions around the muscle fiber. So now we have this new nervous signal, which is going to spread in all directions around this muscle fiber. It's going to spread this way, this way, this way. It's going to even go around to the back this way, around to the back this way. Okay, and this is ultimately what's going to stimulate this muscle to contract. But we kind of got a problem because the sarcolemma only coats the outside of the muscle fiber. It doesn't dive deep into the inside of this cell because that's where all the myofibrils and the sarcomeres are. Well, in order to get this muscle signal deep into the middle of the muscle fiber, we have these special tunnels called T-tubules, which dive deep into the muscle fiber. Okay, so here's a T-tubule here, here's a T-tubule there, and what this allows is this is going to transmit this new nervous signal that's traveling through the sarcolemma to enter in deep into the muscle fiber itself. When that happens, it's going to stimulate these little green shapes that contain orange. That's the sarcoplasmic reticulum inside the muscle fiber. This orange is the same calcium that came into the axon terminal. So there's calcium stored in the sarcoplasmic reticulum. When this signal travels down the T-tubules, it causes the sarcoplasmic reticula to release the calcium. And once that calcium gets released onto the myofibrils, we know what happens next. That calcium binds on a troponin, tells tropomyosin to get out of the way, exposing those actin binding sites on actin, and crossbridge cycling can occur. Okay? So that's really a nutshell of how a neuron is able to stimulate this muscle from contracting. It's all about the release of calcium inside the muscle fiber, and that is caused when the signal from the neuron reaches the muscle fiber.